In this video tutorial, I am going to discuss about the glycolysis. This word glycolysis, it is made up of two words. The first is glycose and second word is lysis. Glycose means sugar and lysis means splitting. So based on this meaning, we can define glycolysis. So glycolysis, it is defined as a glucose is broken down to either pyruvate or lactate. Pyruvate or lactate. This conversion occurs by the sequence of reaction. There are multiple reactions takes place and ultimately it is converted or splitted into pyruvate or lactate. And while doing so, it can also generate ATP. Right? So this is the glycolysis. Now, where does this glycolysis occur? Glycolysis occurs in all the tissues. Right? Because all the tissue requires energy. So the primary purpose of the glycolysis is to provide ATP, or you can also call it as a energy. The second indirect purpose of the glycolysis is that as there are multiple steps, there are multiple intermediates in this reaction, and these intermediates are required by the other metabolic pathway. So this is somewhat less important, but the main importance is to provide ATP in all the tissue. Now, inside the cell, this pathway occurs in the cytoplasm fraction of the cell. Why? Because all the enzymes which are required for conversion of glucose to the pyruvate or lactate, they are present in the cytoplasm. So now let's look at the reactions of glycolysis. So in the previous video, I had explained you that how glucose is transported across the cell membrane. So once glucose is transported into the cytoplasm from the extracellular environment, the first step is that glucose itself gets converted into glucose 6-phosphate. Glucose 6-phosphate. So as you can see over here that one extra phosphate is added to this glucose. Remember, this number denotes that phosphate is bound to the 6th carbon of the glucose. So from where does this phosphate is coming? This phosphate is donated by the ATP. So ATP gives it one phosphate to the glucose, and itself, this ATP itself, it gets converted to ADP. Now, which enzyme will catalyze this reaction? So remember one rule, that whenever ATP is involved in any reaction, that enzyme which catalyzes that reaction is the kinase enzyme. Now, what kind of kinase it will be? So here glucose is a hexose sugar. So we can call this enzyme as a hexokinase, or we can also call it as a glucokinase. So by this method, you can remember the name of enzyme also. Now hexokinase and glucokinase both are different enzymes, but they carry out same reaction, right? Now remember the second rule, that whenever ATP is involved in any reaction and it is catalyzed by some enzyme, at that time for this enzyme, magnesium is a cofactor. Okay, so you don't have to mug up, you can just remember this trick, that whenever ATP is involved, that enzyme is called as a kinase, and this kinase, it requires magnesium as a cofactor. Now coming back to this hexokinase and glucokinase, see, both enzymes, they are different, their structure is different, but still they are carrying out this same reaction. So we can, of course, we can call this as an isoenzyme, yes. But here one notable thing is that although both enzymes are carrying out the same reaction, their entire purpose in the glucose metabolism is totally different. Now, what is their unique feature and how their role is totally different, that I will discuss in the next video, right? Now, the other things to note about this reaction is that this is the irreversible reaction. It is irreversible reaction. That means this same enzyme cannot convert glucose 6-phosphate back to the glucose. There is only one way, right? In case of glycolysis, there are total 3 irreversible reaction, total 3, and this is one of that 3 irreversible reaction. All the irreversible reactions of the glycolysis, they are also regulatory reactions. So this is also a regulatory step. So whenever in the glycolysis we see irreversible step, it is also a regulatory step. Now, what purpose gets served by conversion of this glucose to this glucose 6-phosphate? See, glucose 6-phosphate is too much polar group. It has it is too much polar compound, and along with this polarity, it does not have any transporter in the cell membrane. So, there are no transporters for this glucose 6-phosphate, right. So, because of these two reasons, glucose 6-phosphate, it cannot transported across the cell membrane, right. So, what will happen to this glucose 6-phosphate? Currently, it is in the cytoplasm. So, it cannot cross cell membrane. So, this glucose 6-phosphate, it remains trapped in the cytoplasm. This next thing to note down over here is that the glucose 6-phosphate, it can further undergo glycolysis, but that is not the only fate which glucose 6-phosphate has. Glucose 6-phosphate, of course, it can undergo glycolysis, but it is not the only pathway which this glucose 6-phosphate follows. It can also undergo glycogenesis. It can also undergo glycogenesis. It can also undergo hexose monophosphate pathway, that is HMP pathway, right. So, here we can see that glucose 6-phosphate has a multiple commitment. It is not committed to glycolysis only. It can go to the glycogenesis, it can go to HMP pathway. So, it has a multiple commitment. So, we can say that this is a non-committed step. Non-committed step. Now, generally what happens? If you examine any other metabolic pathway, most of the other metabolic pathway, the first irreversible reaction of that pathway is the committed step, but it is exception over here. This is the first irreversible reaction of the glycolysis, but it is not committed. So, as an exception, we need to remember this. Now, what will happen to this glucose 6-phosphate? This glucose 6-phosphate reversibly, it gets converted to fructose 6-phosphate. Fructose 6-phosphate. Now, see, glucose 6-phosphate and fructose 6-phosphate both are isomers. So, which enzyme will help over here? So, that enzyme is isomerase. Now, what kind of isomerase it is? So, here we can see that it is a hexose plus phosphate. So, it is a phosphohexose. This is also a phosphohexose. So, we call this enzyme as a phosphohexose isomerase. This is the complete name of enzyme. Remember, it is a reversible reaction. What will happen to this fructose 6-phosphate? Fructose 6-phosphate irreversibly gets converted to the fructose 1,6-bisphosphate. Remember, this is the irreversible reaction. Irreversible reaction. Now, as we examine over here, that here only one phosphate is there, whereas here two phosphate groups are there. The second phosphate is bound to the first carbon of the fructose. So, from where does this phosphate is coming? This phosphate is also coming from the ATP. So, ATP loses its phosphate and itself gets converted to ADP. Now, I told you the rule in the first step that whenever ATP is involved in the reaction, we label such enzyme as a kinase. Now, what kind of kinase it is? So, here you can see that it is a fructose which is phosphated one. We call it as a phosphofructose. Here also fructose is phosphated one. So, here also it is phosphofructose, right. So, this enzyme is labeled as a phosphofructokinase. Now, in our body, there are two different types of phosphofructokinase, phosphofructokinase 1 and phosphofructokinase 2. In this case, it is phosphofructokinase 1. Its short form is PFK1. Now, I had told you second rule also, that whenever ATP is involved in any reaction, which cofactor will be required? It will be magnesium, right. So, you do not have to remember, you have to just remember the rules. So, you can automatically recall that which cofactor will be required. I had also told that all the three irreversible steps of the glycolysis, they are also regulatory step also. So, this is also a regulatory step of the glycolysis. Now, the very important thing about this reaction is that this is the slowest reaction of the entire glycolytic pathway. So, the rate of reaction of this step is very, very less. It is the least, it is the slowest of the entire glycolysis pathway, right. So, because of this, it is also called as a rate limiting step. Rate limiting step. Let us further understand why slowest step is called as a rate limiting step. suppose A is converted to B, B is converted to C and C is converted to D. This A to B conversion is catalyzed by the enzyme E1, B to C by enzyme E2 and C to D by the enzyme E3. Let's suppose that the rate of reaction for this enzyme E1 is 100 moles per liter. The rate of reaction by this E2, let's say it is 10 moles per liter. And the rate of reaction by the enzyme E3, let's say it is 150 moles per, not per liter, but per minute. This is also per minute or 150 moles per minute. This is the speed of reaction by this all three enzymes. So, if I want to estimate that what will be the speed of this entire pathway conversion of A to D. So, of course, that will be determined by the slowest step of the reaction, right. So, as this is the slowest step, it is the determining factor for the rate of reaction. That is why we call it as a rate limiting step. Now, the second thing about rate limiting step is that suppose if I want to increase the speed of this entire pathway, that now right now A to D conversion, it is occurring by this 10 moles per minute rate, right. But instead if I want to increase, then what I should do? I should add some factor which can stimulate this enzyme. So, let us say factor X is added and it is stimulating this enzyme, and by this stimulus the speed goes to the 15 moles per minute. So, that means this entire pathway speed will get increased. If I inhibit this, entire pathway speed will get decreased. So, the most effective regulation occurs at this rate limiting step, right. So, of course, in glycolysis there are total 3 regulatory steps, but particular this step, it is going to be the most important regulatory step. Now, once fructose 1,6-bisphosphate is formed, it has only one commitment, that is glycolysis. Fructose 1,6-bisphosphate, it cannot undergo any other pathway. It is committed only to the glucose, only to the glycolysis, and that is the reason this step is also called as a committed step. Committed step. Why? Because once this step occurs, it has undergo glycolysis only. There is no any other fat. That's why it is also called as a committed step. Now, what will happen to this fructose 1,6-bisphosphate? Now, this fructose 1,6-bisphosphate, it is broken down or splitted into 2 compounds. One is dihydroxy acetone phosphate, dihydroxy acetone phosphate, which is short form is DHAP, and the second fragment is the glyceraldehyde 3-phosphate, and its short form is G3P. Now, this is also a reversible reaction, and this reaction is catalyzed by the enzyme aldolase A. In our body, there are total 3 different aldolases: aldolase A, aldolase B and aldolase C. Aldolase A, it is present in all the tissues. Aldolase B, aldolase C, it is present only in the brain, or you can say inside the nerve cell. So, aldolase A and aldolase C both catalyze the same reaction, that is fructose 1,6-bisphosphate to DHAP and G3P. Whereas aldolase B, it is present in liver. It is hepatocyte isozyme of aldolase A, and here it is catalyzing the different reaction. Which different reaction? So, it breaks down fructose 1-phosphate into dihydroxy acetone phosphate and glyceraldehyde. So, this reaction is slightly different than this reaction. Okay. So, this is the reaction which is catalyzed by this aldolase B, and this reaction is important in the dietary fructose metabolism. Right. So, whenever we talk about this step, we have to keep this all 3 forms of aldolase in the mind. Now, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate both are 3 carbon compounds. Okay. See, this fructose 1,6-bisphosphate, it is a 6 carbon compound which is splitted into 2 3 carbon compound. Now, this dihydroxyacetone phosphate and glyceraldehyde 3-phosphate both are isomers of each other and both are interconvertible, and this interconversion of these both isomers are catalyzed by isomerase kind of enzyme, right. So, isomerase enzyme is working over here. Now, what kind of isomerase will be here? So, this is a triose sugar with the phosphate, and this is also a triose phosphate. So, we call this enzyme as a phosphotriose isomerase. See, over here it was a phosphohexose isomerase, as it is interconverting 6 carbon phosphated sugars isomerization, whereas here it is a 3 carbon containing sugars phosphated sugars isomerization, right. Now, glyceraldehyde 3 phosphate can only undergo glycolysis, whereas dihydroxyacetone phosphate, it cannot undergo glycolysis. If this compound has to undergo glycolysis, it has to first get converted into glyceraldehyde 3 phosphate, then only it can follow under glycolysis. So, if we presume that all the carbons of glucose are undergoing glycolysis, then we can safely say that one glucose molecule can give rise to 2 glyceraldehyde 3 phosphate. Now, let us see what is the further story of this glyceraldehyde 3 phosphate. This glyceraldehyde 3 phosphate is reversibly converted into 1,3-bisphosphoglycerate. 1,3-bisphosphoglycerate. So, here 2 important changes are occurring. Here only 1 phosphate was there, whereas here 2 phosphate compounds, 2 phosphate groups are there. And the second important thing is that glyceraldehyde is converted to glycerate. So, let us first address this extra phosphate. Now, from where does this extra phosphate is coming? So, it is unlike previous 2 steps. In the previous steps, that phosphate was coming from the ATP. Here it is not so. Here directly inorganic phosphate, that is also called as a PI, it is incorporated into the glyceraldehyde 3 phosphate and gives rise to this 1,3-bisphospho compound, right. Now, glyceraldehyde is also simultaneously getting converted into the glycerate. So, what is the difference between glyceraldehyde and glycerate? Remember, glyceraldehyde, when it loses 2 hydrogen atoms from its compound, it is converted to glycerate, right. So, we have to remove 2 hydrogen compound. How these 2 hydrogen atoms are getting removed? It is getting removed by this NAD+. NAD+ binds with this glyceraldehyde 3 phosphate, and NAD+ is converted to NADH and plus 1 hydrogen atom free, right. So, this is how this reaction is occurring. Now, as you can see that 2 hydrogens are getting removed. So, what will be the name of this enzyme? Of course, it will be dehydrogenase. It is dehydrogenase. Now, what kind of dehydrogenase it will be? As it is removing 2 hydrogen from the glyceraldehyde 3 phosphate, we call this enzyme as a glyceraldehyde 3 phosphate dehydrogenase. So, this becomes a complete name of this enzyme, and its short form is G3PD. Now, this glyceraldehyde 3 phosphate dehydrogenase, it is inhibited by 2 important toxin compounds. One is iodoacetate, and second one is arsenate. This arsenate is nothing but the pentavalent arsenic. So, we can also call it as a AS5. It is a pentavalent arsenic. Okay, both of these compounds inhibit glyceraldehyde 3 phosphate dehydrogenase. Remember some more thing about arsenate, that arsenate can also inhibit F1 domain of ATP synthase. F1 domain of ATP synthase. Now, the similar looking compound of that arsenate is the arsenite. Arsenite does not inhibit this G3PD enzyme. Rather, this arsenite, it inhibits pyruvate dehydrogenase. This arsenite, it does not inhibit this glyceraldehyde 3 phosphate dehydrogenase. And remember, arsenite is nothing but the trivalent arsenic. So, we can also write it as a AS3. So, don't ever get confused between arsenate and arsenite. Now, let us look that what will happen to this 1,3-bisphosphoglycerate. This 1,3-bisphosphoglycerate, it is reversibly getting converted into 3-phosphoglycerate. 3-phosphoglycerate. So, as you can see over here that here in this compound there is one phosphate less than this 1,3-bisphosphoglycerate. So, where does this phosphate is going? So, that phosphate is received by this ADP and ADP gets converted to the ATP. So, here as you can see that ATP is involved in the reaction. So, by our rule, what enzyme will catalyze this reaction? So, of course, it is going to be a kinase enzyme. And now, what kind of kinase? See, here there is a phosphoglycerate, here also there is a phosphoglycerate. So, we call this enzyme as a phosphoglycerate kinase. This is the complete name of enzyme. And the second rule, that whenever ATP is involved, which cofactor will be required? It will be magnesium ions, right? Now, the very interesting thing to note at here is that the kinase enzyme, whenever it participates in any reaction, almost all the time that reaction is irreversible. But this is the exception. This is the reversible reaction. So, this is one of the rare example of the reaction which is catalyzed by the kinase and still it is a reversible reaction. So, this exception we have to remember. The second thing, it is also an exceptional thing that in our body, generally ATPs are synthesized by the oxidative phosphorylation. Oxidative phosphorylation inside the mitochondria. Most of all the ATPs are synthesized by the oxidative phosphorylation inside the mitochondria. But here in this case, you can see that ATP is directly getting synthesized. This oxidative phosphorylation is not taking place at this level. So, to differentiate it from the oxidative phosphorylation derived ATP, we call this as a substrate level phosphorylation. Substrate level phosphorylation. So, this is very good example of the substrate level phosphorylation, and in biochemistry, you will find very less examples of the substrate level phosphorylation. So, this is one of the very good example of the substrate level phosphorylation. Now, what will happen to this 3-phosphoglycerate? So, 3-phosphoglycerate, it gets mutated and reversibly it is getting converted to 2-phosphoglycerate. 2-phosphoglycerate. Now, why I had used word mutation? See, here the phosphate was on the 3rd carbon and it is getting shifted. Now, the phosphate is on the 2nd carbon of this glycerate. Okay, that's why I had used word that is mutated. So, here which enzyme will be participating? It will be mutase. Now, what kind of mutase? It is a phosphoglycerate mutase. Phosphoglycerate mutase. So, this is the complete name of this enzyme. Now, remember this 3-phosphoglycerate and 2-phosphoglycerate, there is just a shifting of phosphate. So, you can also call both as an isomer. So, this enzyme is an example of isomerase group of enzyme. Now, what will happen to this 2-phosphoglycerate? 2-phosphoglycerate is reversibly converted into high energy compound known as phosphoenol pyruvate. Phosphoenol pyruvate. This is the reversible reaction. This reaction is catalyzed by the enolase, and this is a dehydration reaction. That means water molecules are getting removed. One interesting thing to note about this reaction is that either magnesium or manganese acts as a cofactor for enolase. Any one of these two ions are required for the proper functioning of this enolase, right? And one very important inhibitor of enolase is also there, that this enolase, it gets inhibited by the fluoride. Fluoride can inhibit enolase by unusual competitive inhibition. Unusual competitive inhibition. Now, this inhibition by the fluoride, inhibition of this enolase by the fluoride has several clinical importance. What is it? See, fluoride is added in a blood sample collected for blood glucose estimation. Whenever you collect a blood sample from a patient for the purpose of estimating glucose concentration, at that time we add a fluoride. What is the reason? Why we add fluoride? See, suppose this is the tube in which you had collected blood, and from this blood you want to estimate its glucose concentration. Okay. So what will happen? Inside this blood there will be some cells, okay, RBCs, WBCs, all these different kinds of cells are there, and these cells are continuously utilizing glucose. So what is the problem? Suppose you had received this blood sample in the lab and 2 to 3 hours this sample is not analyzed, right? So what will happen? These cells will be continuously utilizing glucose, so glucose concentration will start decreasing. So as the time passes, the glucose concentration is decreasing. So suppose, let's say after 5 to 6 hours, you started estimating the glucose in concentration. So here you will estimate false low glucose concentration, right? Why? Because these cells are continuously consuming glucose. We have to inhibit this glucose consumption by this cell. So we can add fluoride powder into this solution. So what will happen? This cell's glycolysis will now stop. So now instead of 5 to 6 hours, if you store it for the 10 to 12 hours, it is not a problem. So because of this reason, we are adding fluoride in the blood sample, which is intended for the estimation of blood glucose concentration, right? This is one of the very important reason in this step. Now what will happen to this phosphoenol pyruvate? This phosphoenol pyruvate, it loses the phosphate and is converted into the pyruvate. Now, how this phosphate is removed? This phosphate is donated to the ADP. So, ADP gets the phosphate and itself gets converted to this ATP. Now, this is the irreversible reaction. This is the third irreversible reaction, right? Now, here see, ATP is involved in this process. So, what will be the name of enzyme? Of course, it is going to be the kinase enzyme. Now, what kind of kinase? So, here pyruvate is the important product. So, based on that, name of this enzyme is given, that is pyruvate kinase, and its short form is PK, right? You can also apply second rule, that what cofactor will be required. As ATP is involved in this reaction, magnesium cofactor is required for this reaction. Remember, pyruvate is the end product of aerobic glycolysis, Now, why aerobic glycolysis? We had seen entire pathway of glycolysis. Oxygen is not required at all. Then why this is a product of aerobic glycolysis, end product of aerobic glycolysis? So to understand that, we have to revisit this step, that is conversion of glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. See, here one very important thing is that this NAD+. In our body, the concentration of NAD+ in our cytoplasm, the concentration of NAD+ is approximately 10 raised to minus 5 moles per liter. And if you compare it with the concentration of glucose in the cytoplasm of the cell, it is approximately 10 raised to minus 3 moles per liter. You can see that NAD+ concentration is 10 raised to 2 times lower than the concentration of glucose, or in other words, glucose concentration is 100 times higher than the NAD+ concentration. That means NAD+ concentration is very, very low and glucose concentration is very, very high as compared to NAD+ concentration. So what is there in this difference in the concentration? See, so much of glucose gives rise to so much of glyceraldehyde 3-phosphate, which will consume so much of NAD+. But here we don't have that so much of NAD+. So within a few minutes of glycolysis, all the NAD+ will get used. What will happen once all the NAD+ is used? This glycolysis will stop. Glyceraldehyde 3-phosphate will not be able to convert it into 1,3-bisphosphoglycerate. So what is the solution? Nature had designed our body such a beautiful way that this does not create any problem. Why? Because in our body there is continuous reoxidation of this NADH back to NAD+. How this reoxidation of NAD+ is occurring? In our body, in our cell, there is a mitochondria. This mitochondria, inside this mitochondria, there is an electron transport chain which continuously carrying out oxidative phosphorylation. So what will happen when this NADH passes through this electron transport chain? This extra 2 hydrogen, it combines with this oxygen and gives rise to water molecule, right? And by this way NAD+ is regenerated. Now to just balance out this reaction, I am just adding 1/2 sign in front of this oxygen. So see, in the presence of oxygen and in the presence of mitochondria, this NADH is reoxidized to NAD+. So here oxygen is required, and that's why we call that pyruvate is the end product of aerobic glycolysis, right? But in our body, there are certain tissues which does not have either mitochondria or which does not get enough of the oxygen supply. What are the examples? See, no mitochondria. Which tissue does not have any mitochondria? So the biggest example is the red blood cells. RBCs does not have mitochondria, but still RBCs require glycolysis. Why? Because glycolysis is the primary source of energy, and of course this RBC will require that energy. So even in the absence of mitochondria, RBCs has to generate ATP. Now what are the tissue which lacks oxygen or which has less oxygen supply? So this tissue is a lens, cornea, renal medulla. Normally under physiological condition, all these tissue, they have very less vascular supply, less blood supply, and because of that reason there is a less oxygen. And one more example is that skeletal muscle, generally skeletal muscle has a good blood supply, but when you do exercise, at that time this blood supply is not enough. So exercising skeletal muscle, they also has a less oxygen supply, right? So see, all of this tissue, they require ATP, but there is no mitochondria. Either there is no mitochondria or there is a less oxygen supply. So what will happen? This reoxidation is not taking place. So theoretically this glycolysis cannot take place, but thankfully there is a mechanism in our body, different mechanism in our body, that this NADH is reoxidized. How it is reoxidized? See, this pyruvate, it gets converted, reversibly gets converted into the lactate. And while converting from the pyruvate to lactate, here what will happen? NADH plus H plus, it gets reoxidized to NAD plus. And because of this reaction, whenever there is a mitochondria is absent or less oxygen supply, this reaction will get predominant, lactate will be generated. But here the more important thing is NAD plus is getting regenerated, and this NAD plus will be reutilized at this step, right? So in the presence of oxygen and mitochondria, there is no necessity of conversion of pyruvate to lactate. Why? Because here the mitochondria's electron transport chain is taking very good care of this regeneration of NAD+. But in case any of this is absent, at that time this reaction takes place and it regenerates NAD+. So by regenerating NAD+, this glycolysis will go on and it will keep generating ATPs. Okay. So what enzyme is working over here? So the name of enzyme is lactate dehydrogenase. Lactate dehydrogenase, its short form is LDH. Right. So this reaction becomes very, very important in case of anaerobic metabolism. So lactate, it is the end product of anaerobic glycolysis. Anaerobic glycolysis. Right. So pyruvate, it becomes an end product of aerobic glycolysis, whereas the lactate, it becomes an end product of anaerobic glycolysis. So as you can see, this completes the glycolysis pathway, and we can divide this glycolysis pathway into 3 phases. See, starting from this glucose to ending with this fructose 1,6-bisphosphate. Here during these steps ATP is not generated, rather ATP is getting utilized, right. So, we call this phase as a energy investment phase, energy investment phase. Now, starting from this fructose 1,6-bisphosphate to ending with this splitting, we call it as a splitting phase or lysis phase. And because of this splitting phase or lysis phase, we identify this pathway as a glycolysis. And starting from this splitting till the ending with either pyruvate or lactate, we call it as a energy generation phase, energy generation phase. As you can see that ATP is generated over here at this step as well as at this step. So, energy generation phase, it is occurring for the 2 times. Why? Because each molecule of the glucose, see, each molecule of glucose is giving rise to 2 molecules of glyceraldehyde 3-phosphate. So, that is all for this glycolysis. If you have any query or confusion, please write it down in the comment section below. Thank you.